So welcome to the 22nd session of the International IVF Initiative. We would like to thank Cook Medical for their support of this session. On your screen, we're bringing up a short series of questions. Be sure to click the window and scroll all the way down. There are 4 questions. I am honored and excited to be co-hosting with Kimball Pomeroy. For videos of previous sessions, please go to ivfmeeting.com. You will also find information about upcoming sessions on that same page. Our continued goal is to support the IVF community during the COVID-19 pandemic and provide educational opportunities while bringing our community together. Thanks for logging in, thanks for listening, and please ask questions. My name is Brian LoManto, and I will be co-moderating this session for you. We have an amazing program planned for the next 80 minutes or so on a very special subject that is near and dear to the hearts and minds of our gametes and embryo, osmolality, or osmolarity, depending on who you ask. We have a talented team of panelists who are working behind the scenes to post your questions to our speakers. Today we have 2 guest speakers. Firstly, Dr. Jason Swain, who is author of the seminal review, Optimizing the Culture Environment in the IVF Laboratory, Impact of pH and Buffer Capacity on Gamete and Embryo Quality, which, if you have not read, please do so today. Second, we have Dr. Stephen Mullen, who is unique in the field in that he is first and foremost a cryobiologist. Thank you both for joining us today and sharing your wisdom and experience with us. The importance of a well-monitored pH and osmolality environment cannot be understated. Many physiologic processes heavily rely on these factors. Embryos have the ability to counteract osmotic insult and modulate their internal pH, but these systems are limited. Oocytes and sperm, on the other hand, have extremely limited to no ability to combat these insults, and as such require us to maintain hypervigilance. Our speakers today will dive into this topic and provide a robust understanding and offer objective insight to improve our laboratory conditions and ultimately make our gametes and embryos happy. I encourage you to ask your questions using the Q&A section of the conference service. Some questions will be addressed live and some will be typed in the Q&A section. Please do not use the chat function for questions you want the speakers to answer. Our first speaker is Dr. Jason Swain, who is the Corporate Laboratory Director of the CCRM IVF Laboratory Network. He's responsible for laboratory design, protocol, and procedure implementation, as well as staff training and ongoing quality control monitoring for the network embryology, andrology, and endocrinology laboratories. Jason also oversees quality control of the CCRM donor egg bank. He's an adjunct clinical associate professor in obstetrics and gynecology at Rutgers University, as well as at the University of Minnesota. He completed his Bachelor of Science at Hillsdale College in his native Michigan, his Master of Science in Animal Science at Purdue University, and his PhD in Molecular and Integrative Physiology at the University of Michigan in the lab of Dr. Gary Smith. He then had clinical embryology training with Rusty Poole in San Antonio, Texas, before becoming a clinical associate professor in the Department of OBGYN and IVF Lab Director at the University of Michigan. He's published over 45 peer-reviewed articles, edited and authored several book chapters, and contributed to various other publications within the field of assisted reproduction. His primary research interests include pursuit of methods to improve in vitro embryo culture conditions through reduction of environmental stressors via modification of both the physical and chemical culture environment. Dr. Swain, take it away.
Thank you very much for that introduction, and thank you all for attending this lecture. This is a fantastic series that has been put on for us, and I'm happy to try to contribute and add to our knowledge base. So as discussed, we're really going to be focusing on osmolality and the culture system. So as we know, within our laboratory, one of our main objectives is to optimize embryo development. One way to do that is through minimizing the in vitro stressors that we may impose upon our gametes and cells. If you have improper lab conditions, that can lead to cellular stress and compromise cell development and function. And as we know, there are a variety of potential environmental stressors in the laboratory. And today we're really going to be focused on the one you can see there at the top, osmolality. Well, this is important because despite all of the cutting edge research and innovations in the field, and this webinar series had talked about some of these in the past in terms of artificial intelligence and lab automation, there still really remains a need for basic cell culture understanding and techniques. There is still a large part of what we do that are controlled within the laboratory by the embryologist. We know that commercial IVF media production has been a huge contributor to the improved success in our embryo development and outcomes in our field. This has greatly reduced the variability that we saw when labs were producing varying recipes between different facilities. That being said, there are still things within our control, and importantly, the lab may inadvertently alter properties of culture media, even though they're commercially produced during improper use. And really one of the main routes that this can occur inadvertently in the laboratory is through evaporation. And as we'll discuss in these slides, evaporation can lead to harmful shifts in osmolality, as well as really any solute or substrate concentration in the culture media, but also pH. We'll discuss the pH a little bit at the end of these slides. That being said, there are certain procedures within our laboratories that carry an increased likelihood for potential impact in terms of increased risk of evaporation. And these are procedures that we can pay special attention to if we're trying to combat these potential environmental stressors. One of those procedures is dish preparation, particularly with microdrops, and we'll discuss that. And also uninterrupted culture, certainly an emerging paradigm, especially with the advent of time-lapse incubators. And this is especially apparent in dry incubators. And Dr. Mullen will go into much greater detail in his portion of the lecture on this particular topic. So a good place to start is first to defining what the topic of this discussion, which is osmolality. And a very simple definition is this is the concentration of particles dissolved in solution, or the solute in solution. And this is really determined by an osmometer in milliosmolar per kilogram. And as we heard earlier, there's also osmolarity, which is really just milliosmolar in terms of liter or per volume versus mass. And really the importance of this particular endpoint measure is that this is involved in cell volume control. Without proper osmolality, we can have a potential cell stressor, which could compromise our culture system. And just as a brief review, harkening back to your high school or college chemistry classes, if you'll recall, if you have a hypotonic solution, this is low osmolality, meaning very little solute or particles dissolved in solution in comparison to what's inside the cell. And as a result, water will rush inside the cell and the cell will enlarge or swell. And you can see this visually, probably most readily in the lab when moving through cryoprotectants. When you come out of your vitrification solutions into a thaw solution, the cell will re-expand. And if you don't do that in a controlled fashion, the cell can re-expand or swell too quickly, and you can have cell rupture. And most of us are used to seeing that with freezing of cleavage stage embryos in the past. There's also an isotonic solution, which is no net change or equilibrium in terms of that solute concentration inside the cell and outside in the solution or culture media. And then you have a hypertonic solution, which is a high osmolality solution, meaning high solute concentration or salt concentration. And as a result, water rushes out of the cell and you'll see cell shrinkage. And again, visually, you see this most readily in the laboratory when you move your embryos from a isotonic culture media into a hypertonic equilibrium solution or even higher osmolality vitrification solution where your cell will shrink up. And importantly, as we discussed, we can inadvertently induce a hypertonic situation, high osmolality in the laboratory, primarily through evaporation and really anything that would lead to evaporation. And we'll discuss some of these in greater detail. I do want to say that if you have more interest in osmolality or osmolarity and how our gametes and embryos deal with this in the labs, particularly in relation to formulation of culture media and some of the history, really recommend checking out any number of publications from Jay Boltz in his research laboratory. He is really the expert on this particular subject. And here's just a couple really useful reviews and book chapters that are readily available in human reproduction and other book chapter, but certainly a PubMed search will yield much more from Dr. Baltz and his research group. I highly recommend further reading from these sources. So to try to very briefly summarize a lot of Dr. Baltz's research, the next few slides will give a very brief overview. And as we mentioned, improper osmolality can be a potential cell stressor and impair embryo development. And Dr. Baltz's group showed that very nicely. You can see in the graphs here using mouse embryos that as the osmolality increases, that you see a subsequent decrease in embryo development. And really a quick summary is that if your milliosmolar concentration is much higher than 300 or 310 milliosmolar, you'll start seeing a decline in embryo progression. That being said, transient controlled changes are compatible with development. We know that we can vitrify and warm cells very efficiently. It's really when the osmolality is uncontrolled, changes are uncontrolled through inadvertent lab alterations or for very extended periods of time where we really run into issues. So not surprisingly, given this information, media osmolality is usually somewhere in the range of 260 to 290 milliosmolar. More modern culture media, especially the single-step culture media, tend to be on the lower end in the 260, 265 range. This is much lower than the osmolality that has been measured in the female reproductive tract. So that's probably another topic of conversation that Dr. Baltz covers in some of his various publications. A caveat here is the inclusion of key amino acids is extremely important with proper media in terms of osmolality and the ability of the cell to regulate its volume. Going hand in hand with that, embryos do have mechanisms to regulate their volume, but this is not always without a cost. And so this is, again, a reason why we want to make sure that we're not inadvertently changing the starting osmolality of these culture media, which have been formulated very carefully to optimize embryo development. In terms of some of the mechanisms of how gametes and embryos regulate their volume and deal with osmolality changes, this is a animation that I borrowed from Dr. Baltz. He presented a few years ago. If you have a high osmolality solution, as we mentioned, the cell will shrink. Cells can use inorganic ions through various osmotically stimulated transporters to transport these ions or salts back and forth to regulate volume. This is a very poor long-term regulation mechanism, though. It can compromise subsequent embryo development. A much better long-term solution that's compatible with biochemical function, even at very high concentrations, would be organic osmolytes. And referring back to the last slide, in case of what we deal with in our embryo culture media, is really the amino acids, particularly glycine. And as you'll see, different stages of embryos can use different amino acids to help regulate their volume in response to changes in their osmotic environment. So this again is work from Dr. Baltz's lab, just showing that different stages of embryos have different mechanisms to regulate their volume in response to osmotic stressors. Cleavage stage embryos utilize novel methods for osmoregulation or volume control. These are especially sensitive cell stages, as we know, really sensitive to any number of stressors, osmolality being just one of them. Glycine and glutamine are the main osmolytes used in cleavage stage embryos, and that's why I would argue, and others I assume would agree, that modern culture media really should include glycine, glutamine as well as any other assortment of amino acids. Michelle Lane and David Gardner showed that even very brief exposures to media devoid of amino acids can compromise subsequent development and outcomes. Post-compaction stage embryos, on the other hand, utilize different osmolytes and transporters to regulate their volume. These cell stages are a little bit more robust in terms of their ability to withstand stressors in the environment, and you have higher osmolality needs required for the detrimental effects. But overall, we want to try to, as we mentioned, minimize the amount of stress we put our cells under, even if they are able to deal with a certain amount of stress. So that being said, even if you are using proper culture media with the right assortment of amino acids, there are things that we can do in the laboratory that can change osmolality and have an impact on cell development and function. And as we mentioned, one route of that is evaporation. So just want to prove or illustrate to you that this is a very real phenomenon in the laboratory, and it can happen much more easily than many of us may know. And so here's just an example visually to illustrate this point. This was from a laboratory that did their oocyte retrievals in a sterile laminar flow hood with a flush mounted dissecting scope on a warmed surface. They used 100 millimeter dishes, like many of us do, to search the follicular fluid. Once they identified cumulus oocyte complexes, they would then move them to a 4-well dish with approximately 500 microliters of media inside. And they didn't put oil overlay over those wells because they didn't like the sticky cumulus cells sticking in the interface between the oil and the media, and they sometimes felt like the cumulus cells would float or get stuck in the interface and be hard to get out, or they'd have oil droplets on their cumulus oocyte complexes that would cause them to float once they tried to put them into subsequent culture dishes. So this was their system, and you can see they warm the surface, they try to create environmental stability, they put the lid on the dish. They try to work quickly, but after just 5 minutes at that 37 degrees warm surface, you can clearly see condensation on the lid of this dish, which is a clear representation or indication that evaporation has occurred. And I think most of us would agree that retrievals tend to take longer than 5 minutes. Some can take quite a bit longer. And so the take-home point here is evaporation has occurred. The evaporation is only going to continue as time goes on. And the end result is that culture media is not giving you the same concentration of solutes as it was originally formulated to have due to this evaporation. So end result is the medium properties have changed, and this likely creates a suboptimal growth environment. And certainly some cells are more able to deal with those stressors than others, and you may not see an immediate impact, but ultimately this is a potential stressor that we want to be aware of. So we had mentioned that there are procedures in the laboratory that may be more inclined to encounter evaporation than others. And we just showed one example of oocyte retrievals because these are pretty extended procedures that occur in the laboratory. But another one is preparation of our dishes, particularly microdrops. This is a seemingly benign procedure, often assigned to a junior embryologist or staff member because you're not directly handling gametes and embryos. And the assumption is that there's less risk involved. However, I think we'll show here that this is an extremely important step in the culture process, even if it does seem benign. And if you don't prepare these dishes properly, you can actually create a compromised environment that will be hard to overcome throughout the rest of the time the embryos are in your laboratory. And so that's a study we had done at University of Michigan several years ago in Gary Smith's lab. And very simple design, very simple premise, but I think it still has a very powerful take-home practical point. So what we did is we prepared microdrops under a variety of conditions. We looked at different volumes of microdrops from 10 microliters up to 40. We used a sterile airflow hood, as many of us have in our laboratory, preparing the dishes with the hood on or the hood off. We looked at different surface temperatures in which the dishes were prepared, a warm surface at 37 degrees versus room temperature at around 23 degrees. And then we used a couple different preparation methods for microdrops, what we termed the wash drop method, which is where a microdrop was placed down, oil overlay was then put over the top, the media was removed, and then fresh media replaced the initial drop, versus a standard approach, which was just putting down a microdrop of culture media, covering with oil, and proceeding. These particular dishes were covered with 7 milliliters of mineral oil. These were 60 millimeter dishes. There was around 1.5 to 2 minute exposure time before oil was added. So just to give you a point of reference there. And then we did measure osmolality using the vapor pressure West Core osmometer. And a quick summary of the results can be seen here. We saw a significant impact of the surface temperature with the dishes were prepared, the preparation method of those microdrops, as well as the volume of the microdrop. There was no significant impact in the presence or absence of airflow in the hood. Importantly, there was a significant interaction between the 4 environmental variables. And I think this just helps prove the point of the dramatic impact that this could have. So we had our control medium. Out of the bottle, osmolality was around 290 milliosmolar for this particular media. Under the best preparation conditions, which was the largest microdrop at room temperature with no airflow, using the wash drop prepare method, we saw no significant increase in osmolality, rose up to around 295 or so, maybe 300. However, using the worst conditions, which was the smallest microdrop size on a warm surface with airflow, using the standard microdrop preparation method, we had a significant increase of about 40 milliosmolar. And so in this particular media, that took it up to around 335 milliosmolar or so, which is certainly higher than that 300, 310 mark that we talked about early on. That seems to be the cutoff of where you really start visually seeing compromised embryo development. And there may be more subtle hits to the embryo quality at lower osmolality. And I think a key point here is that we didn't create some artificial stress condition here to get that 40 millimolar increase. We weren't using 1 microliter or some high temperature, some ridiculous amount of time to cause evaporation. I think, certain labs, these are very real world conditions that are used on a regular basis. Now, as we mentioned earlier, this does start kind of thinking about other areas in terms of starting osmolality. If we used a culture media that started out closer to 260 or 265 milliosmolar, we'd be up near that 300 milliosmolar range, which may not visually compromise embryo development. But again, 40 milliosmolar increase certainly indicates that evaporation has occurred and that media is not giving you the same concentrations or characteristics as it was as originally formulated. It's also important to note that media with amino acids, as mentioned, you still may see embryo development. It just may not be optimized if you had lesser amounts of evaporation. Now, that was an older study using, arguably outdated technology in terms of flat 60 millimeter Petri dishes. But the premise and the occurrence is still the same, even with the latest and greatest embryo-specific culture dishes. This is now a few years old from the Vitro Life website technical note from the original embryo slide. But even in dishes prepared specifically for embryos for use in our most advanced time-lapse incubators, evaporation still occur. So you can see a quick summary here. Similar experimental design in terms of dish prep on a heated or unheated surface in the presence or absence of a lid. You can see that after 5 minutes, certain conditions led to more evaporation than others. And then as you had more time, up to 20 minutes, evaporation, not surprisingly, increased. So there may be some differences between dish geometries and configurations, and Dr. Mullins will talk about some of that, but certainly evaporation is occurring in different dishes. And as we'll discuss later on, validating your system is likely important to determine if this is an issue in your laboratory, and if so, how large of an issue. And importantly, we can then come up with strategies to try to minimize that evaporation from occurring. And there are a variety of ways of doing that. Another procedure that we said it likely carries with it increased risk of evaporation and resulting osmolality increase is uninterrupted embryo culture. And this is a review that we published last year in RBM Online that covers this particular topic. And I'm not going to spend too much time on this because Dr. Mullins gives a much better and more in-depth and detailed discussion of these factors in terms of incubation conditions and uninterrupted embryo culture. But I do want to just briefly review some things to help set up some of his discussion points. As we know, uninterrupted culture, really the premise of this is to try to reduce embryo stress through reduced handling, usually entails use of a single step media, try to minimize or reduce the amount of time we're taking the embryos out of the incubator. This is used increasingly more and more because of the emergence of time-lapse incubators and the continued improvement there. And notably, many of these incubators are dry, and that's a very important point when we're talking about evaporation. But really this then poses the question of, is this really that stressful for embryos? And I think, the reviews are mixed on this, like many topics in our field. So you can certainly find publications in the literature that say, yes, uninterrupted embryo culture is better compared to interrupted strategies. However, you can also find publications that say there's no difference. And I would argue that perhaps some of the reasons of why we see this contradiction in the literature is the details, the devil's in the details, these minor differences in experimental design or how the laboratories are culturing these embryos may help determine or explain why some labs see a benefit and some don't. Perhaps some labs are using conditions that are more likely to have evaporation and resulting osmolality increase, media changes, and compromised embryo development. And it's these details that we really need to pay attention to in our role as embryologists to make sure that we're creating a healthy environment for our gametes and embryos. So this is a quick table from that particular review in RBM Online. Most of us are aware of the potential benefits of uninterrupted culture. There's a variety of risks, and for the purpose of this talk, as mentioned, immediate evaporation is a very real risk with the resulting osmolality increase, but also increase in things like pH and other solute concentrations, which I'll mention at the end of this discussion. And so to quickly graphically illustrate this phenomenon, clearly evaporation does occur, and this increases over time. Here's a study that we had published at ASRM a few years ago, looking at 25 microliter microdrops in with 3 milliliters of mineral oil overlay in a standard 35 millimeter Petri dish. We cultured the cells for 1 to 7 days under a dry, modern benchtop incubator condition versus a humidified box incubator that had a water pan in the bottom. And as you can see over time, in the light blue and the dry conditions, osmolality increases. And so this was a way for us to measure over time the occurrence of evaporation. Whereas in the humidified incubator, we really didn't see much of an increase in osmolality, and certainly greater change in osmolality in the dry incubator versus the humidified. And that reached statistical significance after just 72 hours. So there's some important implications here in terms of our culture conditions in the laboratory, time being one of those, humidity being another. And again, this phenomenon exists even with embryo-specific culture dishes and incubators. This is a quick summary of some work that Dean Morbeck published at ESHRA a few years ago now, looking at a control media as well as a Miri time-lapse culture coin, a Primo Vision dish, or an embryo slide. These are embryo-specific slides made for time-lapse incubators, many of which are dry. And as you can see, differing levels of milliosmolar osmolality increase depending on the dish. And this makes sense based on the geometries of the dish and some of the variables that Dr. Mullen will discuss later on. But over time, the osmolality increased, which showed that, again, evaporation is occurring, and this increases over time depending on the culture conditions the dish is exposed to. And it's important to note that this evaporation is occurring under oil. And I think that's something that was interesting to many of us because oil does provide a protective effect, but evaporation is still occurring under oil. And this graph illustrates the type of oil is also another consideration that we should be aware of. Again, very similar conditions, 25 microliter microdrop under 3.5 milliliters of oil, in this case in a 35 millimeter dish, cultured for 6 days in a dry incubator. We looked at the presence or absence of a lid over this time. There was no impact there. But as you can see, using a light mineral oil versus a heavy mineral oil, and we picked oils from Sigma because those were the only oils that we could find that had published densities for them. And this was as far apart as we could find a commercial product for in terms of differences in density, the heavy being only 0.04 grams per milliliter more dense than the light. But there was a significant difference in the amount of evaporation, presumably, that occurred under these oils based on the resulting changes in osmolality that we witnessed. This also brings up another topic, not really suited for this particular lecture, but nomenclature of oil becomes a confusing topic. We looked at 2 commercial products, a so-called heavy oil, which apparently is just a paraffin oil, versus a commercial advertised paraffin oil. We saw no differences there. The amount of evaporation osmolality increase fell somewhere between the Sigma light and heavy mineral oil, but without having published densities on our IVF specific mineral oils, or without having some sort of standard in the nomenclature, it really becomes hard to determine which oil we're using in terms of trying to reduce the amount of evaporation that may be occurring under that oil, whether it's called light oil, mineral oil, heavy oil. It can be a confusing nomenclature system. So I think some potential room for improvement there, especially if we're using dry incubators and uninterrupted culture in this era of increased use of time lapse. So there are some things that we can do in the laboratory to help mitigate this risk of evaporation. One being limit the number of dishes you prepare at one time, especially if you're in a large lab. You really don't want to be laying out 10, 12, 15 or more dishes across the surface of your hood and preparing them by laying down dozens of microdrops before you put oil over. We limit the number of dishes we prepare at one time. We usually work at one patient at a time and split up the number of dishes that we're preparing for that particular patient to make sure that oil overlay is used in a timely fashion if you are using oil overlay. So it's critically important to get that oil over the top of those drops as soon as possible to reduce that chance of evaporation occurring. Be aware of evaporation. So look at your conditions, like the surface temperature of the hood you're using, whether there's airflow or not. I think most of us would argue that we need to have airflow. We want to use sterile preparation conditions. So it's not that we can alleviate all possible sources of evaporation, but we can at least be aware of them and try to minimize those that are more impactful than others. The amount of humidification in the laboratory or whatever the working environment is, whether it be an incubator or isolate, the volumes that you're using, in particular for microdrops, the amount of time used, and really any other factor that can be tied to evaporation. The type of oil, as we just mentioned, and obviously volumes of oil, and Dr. Mullins will talk about that. Klaus Schweimer has done some work with Susan Olds looking at the impact of the amount of oil. Media volumes, as we mentioned, starting osmolality of media could be a consideration depending on your culture system. As we mentioned, make sure you're using media that have the proper composition, meaning the proper assortment of amino acids. But also use of caps and lids. Going back to that scenario of the oocyte retrieval, if you have test tubes of media, it might be a good idea to circularly cap those so they're not sitting there evaporating with the lid off over time. Same with putting things into incubators. Using caps or lids to try to minimize the amount of evaporation that can occur. And then importantly, and I don't think I can stress this enough, and this really applies to anything in the laboratory. If we can measure it, we should probably try to measure it and test our conditions. And so in particular with regard to this lecture, testing our culture conditions so that we know if evaporation and resulting osmolality increase is an issue. So that can be done easily through an osmometer. These can be expensive, but I think most of us at least have colleagues who we might be able to ship samples to. And I think many of our commercial vendor friends in the field, if they've got access to an osmometer, if we would ship them aliquots of media, they would likely be more than happy to test and give us an answer. There's other ways of trying to also determine if evaporation has occurred. And there are some abstracts out there that have used these looking at weight changes versus volume changes of media over time. It's just that you have got to be very careful in terms of the precision and accuracy of your particular devices if using those particular methods. That being said, I'd be remiss if I didn't at least talk a little bit about pH, considering the nice introduction that Brian gave me in the title of the talk. But the reason I'm doing that here is that you could perhaps even use pH measurement as an indicator of possible evaporation in your system. So here's a graph from an abstract we presented a couple years ago at ASRM, and we prepared media using a real-time pH meter in a semi-humidified isolate. In our particular isolates, we have a large water pan, and we get around 40 to 45% relative humidity. And the particular probes on this real-time pH sensor, we applied oil overlay or did not apply oil overlay. And you can see that as time increased, the probe without oil overlay, the pH was drifting up. And this was an indication of evaporation. We could see that visually, but the next day, all the media had dried up. But we were able to measure this with pH, and it certainly makes sense because as media evaporates, pH increases. And so you can see that it doesn't take long for that pH to start creeping up for evaporation to occur even at around 40% humidity. So it doesn't have to be a hot, completely dry incubator for evaporation to occur. And again, we said this likely makes sense. As many of you know, as a brief refresher, the pH of culture media is really set primarily as a balance between the incubator and the culture media formulation, specifically the CO2 levels in our incubator, which we control, and the sodium bicarbonate concentration of the culture media, which our commercial manufacturer set for us. But as evaporation occurs, that concentration of sodium bicarbonate increases as well as any solute in that particular media, it increases. And so in this particular case, as a result of the sodium bicarbonate concentration increasing, there's a subsequent increase in pH. So another way to potentially look at osmolality or evaporation occurring in your laboratory if you don't have access to an osmometer. And there are a variety of ways of measuring pH. Different real-time systems are emerging, blood gas analyzers, and you can still use a tried and true benchtop pH meter. You just may have to do some tweaking to to get it to work depending on your particular culture system and the volumes that you have. But again, the take-home point being here is if we can measure it and help validate our system, especially if implementing a new culture system, if going out and buying a brand new, very expensive time-lapse incubator, you want to make sure that you're not inadvertently creating harmful conditions while trying to create better growth conditions. So to wrap things up, reduced environmental stress during embryo culture is required for optimized development. I think that's a pretty safe statement. Proper measurement of relevant quality control endpoints is useful in validating lab approaches. And so we talked about measuring osmolality in a couple different ways, or measuring pH, to make sure that the conditions that we're using in our laboratory aren't inadvertently creating a stressful growth environment. And care must be taken to avoid unintentionally introducing those stressors. And again, even with all the emerging technology and artificial intelligence and automation talk, there still really is a dire need for basic cell culture and embryology understanding to understand that what you do in the laboratory can still have an impact down the line in terms of embryo development, and in this particular case, evaporation of the culture media. So with that, I'd like to wrap things up and turn it back to our hosts.
Thank you very much, Jason. That was a very well thought out presentation. And I know that your article on osmolality is one of those hallmark articles that every embryologist should read, right along with your friend Rusty Poole's article on pH. And it should be included in the training of all embryologists. You know, you mentioned twice validation, and I think sometimes we forget to validate our processes. We remember the simple validations, but for example, what happens to your HEPES buffered solution as it sits out on the bench, whether it's in at room temperature or whether it's, say, on a microscope with a heating element under it, and it stays at a warmer temperature. Have we really validated all of our methods to follow pH, osmolality, and temperature? And I doubt that we've done that. Otherwise, we would have already discovered what you discovered with your article, that we may be affecting our embryos by changing the osmolality inadvertently. So I think that's really important. Rolf Sen has a question. He wants to know if there's any alternative to oil which can be used kind of to inhibit evaporation during embryo culture. And forgive my dog in the background.
No, I think I'm not aware of anything, and certainly some of our panel of experts may have some additional thoughts on this. But mineral oil, you know, again, it may not prevent evaporation completely, but certainly reduces evaporation. And I think if you've got a high quality sourced product, mineral oil is better than nothing. And we talked a little bit about that early on. And so, you know, I don't know that we can get rid of evaporation completely. But I'm not aware of anything else other than, you know, some of the things Steve will talk about, things we can do to minimize evaporation. But I'm not aware of a product to use in place of mineral oil.
Okay, I've got another question while we're waiting for Steve to get his slides up.
Jason, if you could just unshare your screen there so we can have Stephen bring up his. Thank you.
You know, and that is, what does increased osmolality actually do to cells? What is it? What are the harmful effects that it has on cells?
Yeah, so we had talked, you know, volume control, and you may not be able to visually see that subtle volume control. So as the osmolality increases, the cell's going to want to shrink. That can be a stressor, and we talked about cells have ways of combating that, you know, through the inorganic versus the organic osmolytes. So if they are happening to have to use those salts to regulate their volume, that can create a stressful or more toxic environment. And that's why those organic osmolytes, as Jay Boltz more eloquently discusses, is a better long-term solution. So I don't know that you can visually see the damage. I think it's going to be the unseen damages, you know, intracellularly, where you're going to see the impact. And that could be subtle. You may still get, you know, 49% blastidus formation versus your normal 54%. But I think, you know, osmolality alone may not be enough to completely overwhelm or compromise your system, but it could be one of those dominoes where you have this cumulative effect where if your air quality is slightly off or your temperature is slightly off, and you now have some evaporation going on, you could see your culture system crash. So I don't know that you're going to get an all or nothing response just due to some minor evaporation and osmolality shift, but it's certainly something we want to be aware of to help us in our troubleshooting if we do have a major catastrophic issue in our culture system.
Well, thank you very much, Jason. We really appreciate that presentation. We now would like to go over to our second speaker today, Steve Mullen. Steve Mullen is a team lead and senior scientist in the Reproductive Health Division at Cook Medical. He's served in that position for the last 5 years. He completed his Bachelor of Science degree at the University of Minnesota and his PhD from the University of Missouri at Columbia, where he is also the recipient of the Donald K. Anderson Graduate Research Assistant Award in 2006. After finishing his PhD and a one-year postdoctoral fellowship in the laboratory of the famed cryobiologist, Professor John K. Kritzer, he joined 21st Century Medicine in California to develop a research program in reproductive cryobiology under another famous cryobiologist, and that's Gregory Fahey. In 2011, he was recruited to join the World Egg Bank, where he led the scientific and technical aspects of that company as the science director. Then in 2015, he was recruited to join Cook to serve in his current position. In addition to working for Cook Medical, he's a member of the editorial board for the journals Cryobiology and Cryo Letters, and serves on the executive committee for the Society for Cryobiology. So I'd like to introduce you to Steve Mullen, and I also would like to emphasize that he is not only a cryobiologist, he is an embryologist also. And so I think we'll get some great understanding from this presentation. So it's over to you now, Steve.
You're muted. Oh, there we go.
Okay, there, that's better. Okay, I got different screens up here. I'm trying to figure out which one's going to work. All right, so the second question is, can you see the slideshow, the slide or the slideshow version of the talk? Yes. It's good? Okay. So I'm going to get started here then. First of all, of course, thanks to Jason. He did a fantastic job of organizing what we know to date about this topic. And he's got several great publications out there, and I reiterate what Kim said, that people should be aware of them and essentially have those as part of your knowledge base. I'm going to talk about the topic Jason introduced, and that is the problem we've come to understand over the past couple of years about osmolality changes during culture, and particularly during culture in non-humidified conditions. And I approach things a little bit differently than some people, I imagine. And so hopefully you find this not only interesting, but informative and perhaps useful. I'm trying to develop a mathematical model that will allow us to predict what the osmolality changes would expect to be under different scenarios, which hopefully people will find useful as they determine what methods they are currently using and how they may want to change those methods to overcome or at least reduce this problem. I should say at the outset that I am employed by Cook Medical, which is a manufacturer and distributor of devices, including media and incubators to the IVF industry. And for some reason, it's not advancing either. So give me a second to get this figured out. I've been having all kinds of computer issues today, and I don't understand why. Okay. Okay, so I'm going to outline the presentation here, and I'm going to provide just a brief amount of background information because Jason covered most of this, and this will overlap a little bit of what he discussed. And then I'll talk about some of the preliminary experiments I executed to try and identify some of the key variables that I used to execute a larger experiment to try to develop a multi-factor model that we hopefully will be useful for predicting osmolality changes under different scenarios. I'll talk about the approach to this design, how I developed the model and the analysis of the model, the accuracy of the predictions that I've looked at to date and the interpretation of this model, and finally open it up for questions and perhaps summarize a little bit more if there's time. And so if we talk about the problem at hand, Jason did a really good job of covering this, but we know that our culture systems have evolved over the years, and we know that uninterrupted culture of human embryos has become quite common. The culture is often carried out in microdrops under oil. These microdrops are usually fairly small. 20 to 50 microliters is a common range of drop size. And culture in non-humidified incubators is also becoming common. And the one thing that perhaps has surprised many of us over the past few years is that while we understand oil reduces evaporation quite a lot, it does not actually prevent it. And so there is some evaporation taking place in the embryo culture system over time. And the amount of that evaporation can be surprising given the systems we're working with. So all these factors combined have led us to start to ask some very serious questions, such as how much evaporation actually occurs, and is it enough to be a problem? A lot of people wonder what variables affect the rate of evaporation, and how does the media chemistry change as a result? And finally, the biggest question I think is, are we, or how can we maintain safe culture conditions with the evolution of these new processes? And the graph I show on this slide comes from one of the papers that Jason cited during his presentation. And that just shows one example of many in the literature on how relatively minor increases in osmolality in culture medium can have quite a significant effect on embryo development. And this probably is no surprise to any of us who have been in the field for quite some time. So the big question is, what factors have been suggested to be related to this evaporative loss? And this is just a list that I've come up with looking over the literature. So I don't want to give anyone the impression that I'm the first person to think about this and actually conduct experiments on this. There have been several people, including Jason himself, which is shown in the list in the bottom, to look at different factors associated with this evaporative loss. Of course, humidity levels is one. We know that dry versus humid incubation has an effect. The type of oil, including the density and viscosity as properties of the oil, have been suggested to be a significant factor. The initial oil humidity, which is an interesting thought, which has been suggested by some to be a possible way to mitigate this problem. Most of the literature that I've seen suggests that trying to pre-humidify oil really has no effect. And I won't show slides for the experiment I conducted, but because it was essentially a negative study. But what I showed was that if you pre-incubate oil in a humidified incubator for up to 5 days and then use it in a dry incubator setting, there's essentially no effect of that process on the rate of osmolality increase. Of course, the volume of the drops and our surface area associated with the drops of media can have an effect, and that is related to the geometry of the media in the dish. For example, if it's in microdrop form versus in a cylindrical form in a well, for example. And also the surface area and volume are associated, and I'll talk about that during an upcoming slide. People have speculated that the oil depth above drops can have an effect, and there's been some experimental evidence to confirm this. And finally, it's been discussed, and as Jason showed, that the type of incubator or the manufacturer of the incubator, shouldn't say the manufacturer, but the type of incubator manufactured and used can have an effect on this process as well. And so what I'm going to do is now transition into discussion of some of the preliminary experiments that I conducted to confirm some of what has been published in the literature already, and to try to get an estimate of the quantitative effects of these different factors. And the first experiment I'm going to discuss is, what is the effect on osmolality rise of the different volumes of microdrops in culture? And in all the experiments I will talk about today, were conducted in a mink incubator that had an empty humidification flask, so we could create a non-humidified incubator setting. And as I'll mention in a minute, in that condition at 37 degrees, the humidification, the relative humidity of that system is about 10%. And the second thing is, unless otherwise stated, especially in these early studies with microdrops, I was using 3 mils of oil overlay in a 35 millimeter dish. And I would conduct these studies over 7 days and sample over different days across that 7-day time period to determine what the change in osmolality was. And what I have done, instead of focusing on specific osmolality values on a certain day, for example, I've looked at the rate of change over time, as you can see in this graph here. as you start off with an osmolality somewhere in our system, about 285 to 290 milliosmol, and looking at how that change in osmolality occurs over time, and then calculating the rate of change simply by performing a linear regression on the data. And in all these preliminary experiments I looked at, there was, in many of them, I should say, there was a linear relationship between the time and the change in osmolality. So if we look at the different slopes of these regression lines, that indicates the rate of change, and I've simply calculated the slope coefficient and the 95% confidence interval for that coefficient under these conditions here of 30, 40, and 50 microliter drop volumes. And I saw what I was expecting. I expected to see a smaller drop volume would have a higher rate of osmolality change. And I was a bit surprised to find out that the rate of change between 40 and 50 microliter drops was fairly similar. And the reason why I was surprised by that is because I was expecting there to be a relationship between drop volume and osmolality change. And also, during the early part of this study, I was using a rather crude method to estimate the surface area associated with these drops. And because I realized it was a rather crude method, that was always bothering me in the back of my head. And so I started to ask around a few people and describe what I was trying to achieve. And one of my colleagues at Cook, Jonathan Bennett, who was one of the engineers, he told me about this microscope that Cook has in their engineering testing laboratory, which is shown here. And it has an arm that can rotate around a 180-degree half circle, which allows us to take pictures of microdrops from a side view, which is shown in this left-hand slide. And so what I've shown in the right-hand part of this picture on the left is that we can use quantitative image analysis to measure things such as the arc length of a drop, for example, shown here. And also we can use that to measure the height of the drop in the middle, for example. And then that information can actually be used to determine, or at least estimate, a relationship that would describe a functional relationship between an X and Y variable of this particular arc. And not being a mathematician, but I have friends who are. I got a good friend up in Canada, James Benson, who's a great mathematician and cryobiologist. And whenever I have a challenging math problem, I often send him a question and he often helps me find an answer. And he reminded me of this method of integral calculus that can be used to estimate surface areas if a person knows an XY relationship or a functional relationship. And with the information we got from these image analysis, we can actually estimate a functional relationship that describes the arc of that drop, and using this integral calculus method, we can estimate the surface area. And it gives a much more precise estimate than the crude method I was using earlier. And so I executed that experiment and determined the surface areas associated with 20 through 50 microliter drops. And I'll show that data in this table. And not surprisingly, the surface areas go up as the drop volume goes up. But what's interesting and helps to explain the data I saw and showed in the previous slide is that the surface area to volume ratio actually sort of, it goes down as the drop volume goes up. And at least for 40 and 50 microliter volumes, the surface area to volume ratio is essentially the same. And so that lends credence to the idea that it's not necessarily the volume of the drop, but it's actually the surface area relative to the total volume that affects the osmolality change. And that's not too surprising. In fact, it's pretty much expected based upon what we think the system, how we think the system will behave. And so with this information now, I have a good estimate of the surface area to volume ratio of microdrops, and I can use that as part of an experimental procedure. So the next experiment I executed was looking at the effect of different oils from different manufacturers on the rate of osmolality change. And I'm not so interested in a specific brand of oil, but more interested in the properties of the oil. And I'll talk about that in the next slide. But the way the experiment was set up is described in this bullet point list here. Again, 30 microliter medium drops, 3 mils of oil, 35 millimeter dishes. We tested 9 different oil types from 7 manufacturers. And we cultured in the dry incubator, as I mentioned, at about 10% relative humidity. And when you look at the graph, you'll notice there's a time 0, and that just indicates the time that the dishes were placed into the dry environment of the incubator. And so this graph shows the rise in osmolality over time with the different oils. indicated by either dashed lines or solid lines here. And what you can see is that there's quite a bit of there's quite a big difference in the slopes of these lines depending upon the oil type. And again, when you look at the slope itself and estimate the osmolality in the different, estimate the slope parameters in osmolality change with these different oils, you can see that there's quite a difference in the rate of this change, with the best oil, if you will, having an osmolality change of about 3.8 milliosmolal per day, and the worst oil having an osmolality change of about 8 milliosmolal per day. So you get about a twofold effect depending upon the type of oil you're using. And I included this data point down here to show what we would expect to see in a humidified setting under similar circumstances. And I didn't show all the information about this particular experiment, but I selected oils that range this, that span the range, if you will, of these variables. And all of the data suggested that under humid conditions, there's really no effect of oil type. And so I combined those data to determine what the expected rate of osmolality change would be in a humid condition. And it turns out on a daily basis, excuse me, or 24-hour period, you would expect about a 1.56 milliosmolal change versus the best case scenario in a dry environment, which is about 3.8 milliosmolal change. And so this helps us to understand that when it comes to oils, even the best oil available cannot compensate for the fact that you're culturing in a dry environment. So I mentioned I'm interested more in the characteristics of the oil and not oil brands themselves. And so my colleague Sean Tanner at Cook Research up in West Lafayette executed experiments to measure the viscosity of the oils. He has a nice instrument up there to do that. And I determined the density of the oils, both of these at 37 degrees C. And just looking at the relationship between viscosity and change in osmolality and density and change in osmolality, we see that both of these variables have a predictive effect on the rate of change. And you can see that this appears to be a nonlinear relationship as shown by this curve fit here. But for density, the relationship appears to be quite strongly linear with the R-squared value of 0.9055. And when you perform a multiple regression analysis, you quickly learn that both of these factors are not independent predictors of osmolality change. And partly strongly because they're correlated with each other. And so when you want to try to model the properties of oil, you have to choose one or the other. You wouldn't want to choose both because they won't both contribute independently. And because of the fact that this is a simpler relationship and also a stronger correlation, I chose to move forward with oil density as a property of the different oil to include in the model. So the final experiment I did was looking at the effect of oil volume on the rate of change in osmolality. And this is the first experiment that I'll talk about where I used a plate format instead of a drop format. And the reason I chose to do that moving forward is because, as I mentioned earlier, the surface area to volume ratio in drops is not so easy to control. Whereas when you have a well format like this, you have a fixed geometry and therefore the surface area is fixed. And so you can control the surface area to volume ratio much easier simply by adjusting the volume. And so I wasn't so much interested in surface area to volume ratio in this experiment, but I did start to use that format so I could have that as a means to control surface area to volume. Okay, so what we saw here was that between 40 and 50 microliter drops of media, as oil volume goes down, rate of osmolality change goes up. And that's not something that's too surprising. There does seem to be a bit of a split between the 40 and 50 microliter volumes here. And I think that can simply be attributed to the fact that there's no longer the same surface area to volume ratio between these two volumes of media when you're in a 96 well plate format. Okay, so I did conduct a few other experiments I won't talk about today and ruled out a few other variables that would be important to include in a model. And so I moved forward with this three-factor model to try and understand how these three factors of surface area to volume ratio of culture medium, the density of oil, and the thickness of the oil layer interact to affect the rate of osmolality change in non-humidified culture conditions. And so I chose ranges of these variables shown in this first bullet point that are practical and also practical in a sense that that's a common range in which people use in the laboratory. And the range of surface area volume I estimated based upon the different type of dishes people tend to use. And so these surface area volume ratios should cover a wide range of scenarios that people are using in the laboratory. Again, I use a 96-well plate format, and I could adjust the volume and achieve a surface area to volume ratio as shown here. We always use a Sydney IVF cleavage medium for these experiments simply because it's what we have easily available to us. And I don't think the results are really only limited to that particular medium. Mink incubator, of course, as I mentioned, used at 37 degrees C with a relative humidity of 10%. In order to try to minimize unwanted variable influence, I randomized a run order as well as a sampling order in each block of the experiment and measure the osmolality, as I talked about, over a 7-day period with sampling of a single well per day. And I use a Westcore vapor pressure osmometer similar to what Jason uses. Utilizing response surface methodology, I could execute this experiment and analyze the data. And there's a really good software program called Design Expert that's based out of a company in Minneapolis called StatEase. And I really like this software. So if anybody's interested in either looking for a software package or using one, I certainly would give this a look. So for the people out there who are familiar with response surface methods, I started with this face-centered central composite design. It was essentially a 3x3 design with the points being at the extremes of these ranges here and one point in the middle. And then after the first block, I executed additional blocks of data with 19 to 24 runs per block, sampling within different areas within the ranges associated with these parameters. And in the end, I had executed 107 different independent combinations of these variables to produce the three-dimensional plots that I'll talk about. So I'll go through this a little bit quickly. I don't think it would be good for me to skip this, but nevertheless, for those of you who aren't familiar with the details, don't worry about it so much, but I'll just try to walk you through a little bit of it. And so with response surface methodology, you estimate surfaces with polynomial equations. And I looked at the quadratic model, a cubic model, and a quartic model in this study. And it was pretty clear to me that the quadratic model actually fit the data pretty well. But in certain parts of the experimental range, the cubic model was a better fit. And you can see how criteria such as the Akaike information criteria has quite a bit of a change between the quadratic and the cubic model. And there's not a very big change going to the quartic model from the cubic model. And in addition, when I looked at how the model was predicting the response with the quartic model, it strongly suggested to me that was an overfit of the data. And so I stuck with the full cubic model and then performed a backward selection process to eliminate any combination of factors that were not significant predictors of the outcome. And just as a note, in order to perform this within the confines of the model assumptions, I had to perform transformations on the data. And so a little bit more information about the model. 7 of the terms from the full cubic model were eliminated. None had to be returned to achieve model hierarchy. And all of the terms here essentially listed in this part of the table have very strongly significant effects on the prediction of the outcome. As I mentioned before, I had to do a square root transformation of the data for the cubic model. And so if a person were to try and calculate expected osmolality changes under different scenarios, the equation will give you the square root of the osmolality change. So you need to square that in order to get the true osmolality change. And so the equation that describes this relationship is shown here. I wanted to show this in case any of you are interested in trying to do a little programming for yourself. But this shows the different terms of the equation, and the entire thing needs to be squared, as I just mentioned, to get the final variable of osmolality per hour. Now, in this case, X equals the surface area to volume ratio, Y equals the height of oil in millimeters, and Z would equal the density of oil. So it's this equation that we use to try to estimate how well the model predicts the actual experimental data, and then to try to see if we could extrapolate beyond the experiment and get useful predictions in different scenarios. And so what I'd like to do is just go through the 3 of the graphs that the model, the software gives to you in order to determine how well the model fits the data. And this particular graph shows the expected response of the variables oil height and surface area to volume ratio when the oil density is at its lowest factor. And I should point out that the range of values on these axes are coded, and that's based upon how the response surface methodology is executed. All you need to know is that the -1 indicates the lowest value in the range, and the +1 indicates the highest value in the range. And the factors between those would be proportional within the range itself. And so when the oil density is lowest, we see quite a dramatic difference in osmolality change over time, which is indicated by the height from the bottom of this graph to the surface. So the mesh surface indicates what the model predicted values are, and the individual dots are the values determined from the experiment. And you can see a couple different things stand out immediately by looking at this graph. One of which is there's a very steep slope as you go from the left front to the right rear of this graph, which indicates that as surface area to volume ratio goes up and oil height goes down, there's a very strong response in that scenario. The other thing that is pretty clear initially is that at the front part of this surface, as a surface area to volume is relatively low, oil height has a very little effect. So when you have surface area to volume ratios around what would be in real terms of about 0.05, sorry, oil height has very little effect on the rate of osmolality change. But as you go from the smallest to the largest surface area to volume ratio, thicker oil heights have an effect of diminishing the rate of osmolality rise compared to when you have a thinner layer of oil. Now, if we look at how the oil density affects the system, I'll show these last 2 graphs. And this graph shows when we have the oil density at an intermediate value and oil density at the highest value. And the part of the graph that has the biggest effect is this far right corner, as you can see. As the oil density gets higher, the rate of osmolality change on that far right back corner of the graph goes down quite a bit. In fact, it gets to about half the value. And so what that indicates is that if you have a high surface area to volume ratio, high oil density can help to mitigate the problem of the osmolality change over time. There's not that much effect with oil. Oil density does not have much of an effect when you have a low surface area to volume ratio and when you have a thick oil layer. And so you can see that the change that occurs predominantly between these 2 graphs happens when you have a high surface area to volume ratio and a low oil height. Okay? So now we have a mathematical equation that can help explain what we would expect the change in osmolality to be under many different scenarios. And one important thing to consider is how does this model predict what we would expect to see in microdrop cultures? Because I was not using a microdrop culture system when I was conducting the experiment. But I did happen to have data that I collected from a different experiment with microdrop culture in a non-humidified environment. And so what we can do is we can look at those data and determine how well the model predicts the outcome of those experiments. And this column here, indicated by the red, shows the actual experimental response in milliosmolal per hour that was determined when we used these factors of these different variables, these factor levels of these different variables. And this column indicates what the model would predict the actual response to be. And what you can see is that there's a fairly good concordance between these numbers in these 2 columns. These columns over here represent the confidence limits of the mean for the prediction and the prediction limits for the overall populations. And with the exception of 1 data point, all of the actual responses fall within the upper and lower confidence intervals of the expected mean response. And for the one that doesn't, it falls well within the prediction limits here. And so what this tells me is that the model has pretty good predictive qualities for microdrop scenarios, even though I didn't use microdrops during the experimental period to develop the model. Well, the final graph I want to conclude with is shown here. And this shows a comparison between what we would expect to see in humidified versus non-humidified conditions. We've seen the red surface already from the previous slides. And this is when we have oil at an intermediate density. And the black surface indicates what we would expect to see across different regions of this graph when we are working in a humidified system, okay? And the one thing you can tell, which we've talked about, is under non-humidified conditions, the expected rise in osmolality is only comparable to the humidified conditions when you have a relatively, this should actually be, surface area to volume ratio is low. I have a typo here, so please forgive me. This should be low. So when the surface area to volume ratio is low, under non-humidified conditions, the expected response between humidified and non-humidified overlap in this part of the curve. Okay, and so what that tells us is that under microdrop conditions, which has a surface area to volume ratio back in this part of the graph, we don't ever see a scenario where the humidified and non-humidified conditions are similar. And that's telling us that it may very well be impossible to achieve a osmolality change that's equal to what you would expect to see in a humidified conditions with different levels of variables if you have a non-humidified setting. And so that's an important point to consider. And this really highlights what Jason mentioned at the end of his talk, that we really need to understand the quality control settings of our culture system and how we may be changing, even unknowingly, different parameters within our culture system. And if we can understand better what we would expect to see and using an approach like I've discussed here, we might be able to get a better handle on what the changes in different variables could be under our settings. And that may help us to guide us in directions of changing our system in certain ways that could help us to achieve better results with our embryo culture. So that's an end of the presentation I want to give to you today. There are a number of individuals that have helped me along the way, particularly the people I work with in the IVF Research Lab, as shown in this top bullet point here. Cassie Shelton, she has about 10 years of clinical embryology experience, and she's come on and been a senior scientist and the laboratory director for, our laboratory manager, I should say, for our lab. So she does a lot of the kind of work that a lot of clinical embryologists do to try and keep our quality control settings in the research lab at a high level. James Benson is my friend and colleague up at the University of Saskatchewan, who's a mathematician and a cryobiologist, who helped point me in the direction of using the integral calculus method for determining the surface area of microdrops. AJ McKenchny is a statistician at Cook Medical who helped confirm that I was on the right track with the response surface methodology. And Brian Woodard and Elizabeth Brown are two of the people I interact with on a routine basis at Cook Medical. And without their continued support of what I do, I wouldn't be able to do the kind of work I enjoy doing. So thanks to them as well. So if anybody has any questions directly for me, here's my email address. I hope I'm not opening myself up to a couple long weekends of responding to emails, but I will do that. If you want any more information that I can provide to you, feel free to send me an email. I'll see if I can help out. And with that, I'll turn it back over to the moderators of the session.
Thank you so much, Stephen. That was very intriguing. I don't know why I've always assumed if you put oil on top of a liquid, it was the equivalent of putting a lid on it, that it was going to stop any sort of gas exchange. But it obviously does not. And especially when, for oil to work as it does, it has to be less dense than water.
Right. I think you highlighted a good point about when you said gas exchange. What I suspect is happening is that individual water molecules are breaking free from the surface and diffusing through the oil and diffusing out of the system. So I think we might be in a situation where the water is acting more like a gas because it is entering a gas phase essentially. So it may be that that's why we're seeing what we're seeing. And I think it's the reason why it's surprising is because everybody just assumes, like you said, that the oil is covering the water and the water doesn't move through the oil because that's just the nature of oil. And we're learning now that that's the nature of oil to a limited degree. And it may be enough of a problem that we could be affecting our embryos negatively.
Yeah, that's very eye-opening. I really appreciate the time you spent in that, and I can't wait until you finally publish the entire material together, I'm sure, with additional experiments. We're now going to open up this series to questions. Questions either for Steve or for Jason. And I know that we have one question from Devajani Mahapatra. How are osmolality, pH, and temperature different in relationship to overlying oil? This is a tough one, guys. In closed culture systems and open culture systems. So for either Jason or Steve.
Well, you may want to repeat that for one thing, and then define what open culture and closed culture systems means.
Yeah, I don't know that myself, but I'll try make a stab at it. How are osmolarity, pH, and temperature different in relationship to overlying oil? And I imagine by a closed and open culture system, they're referring to what we call a dry or a humidified system.
Okay.
I always interpreted closed and open versus is with or without oil overlay. So it's kind of unclear. I mean, the dynamics really are the same, and you'd want the same endpoint. So if your pH, if your endpoint pH is 7.25 in a closed system, that should be your goal pH in an open system. And same with osmolality. If you want 265 as your osmolality in open, it should be that way in closed. Same with temperature. So I don't know that any of those endpoints vary between an open and a closed system. Now, that being said, an open system, if I'm interpreting correctly, which means no oil overlay, you would have the potential for more evaporation, faster gas exchange. So you may have faster pH equilibration. You may have faster evaporation. Right. But in terms of the end result of what I want to expose my embryos to, there should be no difference. And again, my history certainly isn't as extensive as some of yours. But, when I heard of open culture in the past, it was, we don't want mineral oil because of the toxicity concerns and issues we historically had with mineral oil prior to our commercial manufacturers kind of pre-screening that for us. So, I guess in quick summary, there shouldn't be that many differences, in terms of the environment we're trying to present to our embryos. If I'm interpreting open versus closed correctly, you just may have some differences in timing in terms of dynamics for how fast evaporation occurs or how fast pH equilibration occurs. But, the end goal should be the same regardless of open or closed.
Right.
And I think you've interpreted it correctly, because the second part of that question was, do any labs practice open culture systems? Mm-hmm. So you probably interpreted it correctly, not the way that I said. I think, though, you point out an important point. If you want to know what's happening in your system, measure it. Mm-hmm. You should determine in your own system. You should validate it. What's going on with osmolarity, pH, and temperature in whatever system you're using.
Yep. Yeah, I think too many people focus on pH and temperature. And I think what we just learned here is that osmolality is probably just as important, and we should add that into our daily routines, right, or at least our ongoing QA systems.
And it's going to affect the pH.
And we'd even said, at least validate initially, right? Because it's expensive to have an osmometer, and people have had the same discussion with pH. How often do you measure? People like Joe Conahan measure a lot. Other people may measure, every new lot of media, or every new gas tank if they use premixed gases. So at least if you validate initially, hey, if I'm confident that my microdrops are always this size or this shape, as Steve, clearly pointed out, that, a bigger microdrop may flatten out, and there could be different dynamics involved there. But if you're confident that your system is consistent and you always use 3.5 mLs of media, and you always use 30 microliters, and the dome is always, shaped like this because of the surface nature of the dish. If you do it initially, with reasonable expectations, you could expect it to be consistent, but not always. I think most of us have seen that. Incubators go awry, conditions change, and that's why some sort of ongoing measurement is usually advised, but at least initially, to see if you've got a problem or not.
I agree.
Definitely.
Yeah, I think that the rate of change is really the biggest factor. As I showed here, and as many others have shown with pH, that it will change if you take the lid dish out of the incubator, for example, but the oil really retards the rate of that change. And I think that's what the oil does. That's how it helps.
Yeah, I think a lot of people might be sitting here saying, oh, shoot, I do my retrievals with a center well dish or a 3.5 millimeter dish, and I don't put oil over that. Should I be doing that?
Well,
I think the answer might be yes.
You know, if you're in and out of it, you don't have a lid on it, so you don't visually see that evaporation. If you put that lid on that, I think a lot of people would be shocked. Oh, wait, there's condensation here after a pretty short amount of time.
Yeah. Absolutely. So let's get another question here. We'll do one more. I think we're getting a little low on time. So it's from Diego Escura. It says, isn't it easier to protect your embryos by adding humidity to the culture system instead of doing any type of calculation for dry culture systems? Human oviduct environment is not dry, so why use a dry environment? I think that's on everybody's mind.
The embryo isn't in a dry environment. It's in media,
Right.
so it's 100% humidified.
Right.
And I would argue that maybe the female reproductive tract isn't 100% humidified, staying in ocean media. But I mean, the reality is the logistic aspect is the incubators that we can purchase, these benchtop fast recovering incubators, are largely dry. And we certainly don't want to go back and add humidity to these and muck up the inner components. And there are incubators like the Mink and others that are humidified, but sometimes we're kind of handcuffed or pigeonholed by the technology that we have at our disposal. The nice thing is, as Steve clearly showed with his numerous variables, is we can do things to mitigate that risk, even in a dry environment. You can modify your drop volumes in geometries and add more oil and do different things to reduce that risk.
Yeah, that really was the driving force behind why I chose to do this, because people were asking those questions. If we want to use a dry incubator, but we know this problem exists, is there anything we can do to our system that doesn't involve changing incubators to reduce or eliminate the problem? And so that's what I tried to look at that and determine if there is.
Well, and I think it's good to remember why some people prefer dry incubators is depending on where your lab is, part of the world, contamination, right? Humidified incubators can have contamination. The dry incubator helps address that particular issue. So it's a trade-off. And the nice thing is it's probably easier to address evaporation than contamination, depending on the source of the contamination.
I think so.
This has been an excellent discussion, excellent presentations. I would like to stay here another hour, to be honest with you. Jacques Cohen has a question that I think would be great to close out on, and I'll present it in maybe a very, very short discussion, but it'll give us something to think about. Oil and water don't mix, yet in our culture system, they seem to mix somehow. Culture medium is not water, even though it consists of more than 99% water molecules. Something does not add up. There must be one or more emulsifiers present in culture media, allowing water to permeate through oil. Proteins like albumin have emulsifying properties, and maybe some of the salts do as well. Any thoughts?
I think it gets to what is meant by oil and water don't mix. I don't think, this is certainly not the first time people have noticed that water can get into oil. When I was at Purdue and Jason was at Purdue, I blew a head gasket in one of my cars and the oil turned into essentially a chocolate milkshake. Or not the oil, but the coolant. So oil and water don't mix in the way we usually think about it. But I also think he's probably correct in that there may be components in the media that accelerate or enhance or allow the water to get into the oil easier than it would if it was just a pure water system. Now, it would be, as he mentioned, pretty easy to test that. We could simply just use pure water and repeat the type of experiments I did and determine if the change in osmolality differed as a result. And I might just throw that in when I try my next set of experiments. And that would help to understand that. And maybe that is one other factor that can be changed in our culture systems to try and mitigate this problem. And
did your media have bovine serum albumin or some other protein source?
Yep, it had albumin in it. It's the standard Sydney IVF cleavage medium you can purchase from Cook.
Sorry, I forgot. We don't use bovine serum albumin anymore. Human albumin.
It's human albumin. It's not BSA. That's right.
Yeah. Jason was laughing at me.
Yep.
Any other comments, Jason?
No, just that, yeah, same thing. We used a complex protein with globulins in our particular studies. Steve's got some other studies planned, it sounds like. But, Steve and I had both also done some experiments where we tried to, quote unquote, saturate oil by pre-washing it, at least in my case, with culture media, with protein, and that had no impact on the rate of evaporation too. So I think that is something to think of in designing those new experiments that, we at least, and I don't think Steve was either, able to saturate oil. So I don't think it's that the oil is somehow, absorbing the water away from the media. I think it truly is, an evaporation or an emulsification type.
Yeah. Yeah, some of the experiments I didn't discuss involve changing the number of drops in a dish to see if we could answer the problem, or the question is oil acting as a sink for water, or is it just acting as a conduit to get to the atmosphere beyond it? And it seems from the studies Jason has done, what I showed in our lab and others, that oil is really not acting as a sink, because when you put the oil in an incubator, if this phenomenon of water getting into the oil is happening, it seems like it would happen in that scenario too. But when you put that then into a dry incubator, I think the water evaporates from that oil very quickly. And so there's really no discernible effect of that.
All right. Well, I'd like to thank everyone, especially our speakers, for sharing their time and their expertise with us. I'd also like to thank our team of panelists who work in the background to make sure questions are prepared and answered and do a lot of the ordering that helps this to run smoothly.
Kim, would you like to take a quick look at those poll results before we sign off?
Brian.
All right.
The poll results. These were to get a lot of us thinking about things before the lectures.
Yeah, so the first question is an oil overlay a good inhibitor of evaporation such that osmolality does not increase over several days of culture? Most of you said yes. Some said no. A few said they don't know. I think we might have learned here that it is good, but there are certain factors that we need to look at: drop size, amount of oil. Anybody have anything to add here?
I think the key to that question is
don't have an osmometer.
Does not increase over several days. I think that's the key.
Yeah. I think we did learn that it actually does increase, but that increase can be different based on your culture system.
Right.
Most labs don't have an osmometer. I feel like we all should be rethinking that. Oil density, as Steve elegantly pointed out, does affect evaporation of media. And this question of pre-humidifying oil, Jason, I think you just spoke a little bit to that. Do you have anything to add about that?
No, I think Steve is the one who I believe did the studies trying to pre-humidify it in a humidified incubator. We tried doing that by washing, and I think we both came to the same conclusion, that it doesn't seem to really have an impact.
Well, there we go. Thanks very much. Go ahead, Kim. Over to you.
Thanks a lot for reminding me about that, Brian. That's why there's two of us here. I'd like to thank especially the founders and the ever-present beings that are shuttling this enterprise into glorious future. Peter Naj, Thomas Elliott, Shaista Shadruddin, and Marianne Savatu. Finally, don't forget, join us for our next session. It will be Laboratory, Planning, Maintenance, and Engineering Now and in the Future. It will be hosted by Jacques Cohen and Giles Palmer. And the speakers for that session are Jacques Cohen, Antonia Gilligan, Chip Calcagni, and Tucker Matthews. Sorry, Chip, if I murdered your name. I'd like to thank each and every one of you for joining us today. Without your participation, this would not be possible, and it's this interaction that makes this sort of forum very good. Thanks so much.
Thanks, everybody.